Good morning, all. Today we are studying on the factors affecting enzyme activity. Up till we see the what is the enzyme classification, properties of enzymes, uses. Now we study the which factor is affect on the enzyme activities. Enzyme is the more important in life. Enzymes are highly specific for the types of reaction they catalyze and for their substrate. Enzymes with highest specificity and accuracy are involved in copying and expression of genomes. These enzymes have a proofreading function. Which enzyme? For example, DNA polymerase. This is the enzyme which have a proofreading function, having a specificity and accuracy are involved in the copying and expression of the genome. Mostly the mechanism of enzyme action is that the enzymatic reaction takes place by binding the substrate with the active site of the enzyme molecule by several weak bonds. When the enzymatic reactions occur, the formation of enzyme substrate complex firstly, when the enzyme substrate complex form by active site of the substrate and the enzymes. Then the catalytic reaction is occurred between the enzyme and the enzyme substrate complex is subsequently converted into the product. The converted into the product, when the product is formed, then the enzyme will be free. We see in this graph, you all look at that. This graph having a two graphs in a red line and in blue line. In blue line, which is done with the enzymes, and in the red line graph is the reaction which is done without the enzyme. So, some reactions without enzymes occurred, having a more energy to complete the reaction. In another case, the when enzyme, what is the role of enzyme can increase the rate of reaction. Then in this blue line, you all see the activation energy with the enzyme is low. So with enzyme the completion of reaction is faster. Okay, then we see the factors affecting reaction velocity. Temperature. First is the temperature. Each enzyme has an optimum temperature at which it works best. Temperature rises above certain point, it loses its three-dimensional structure. The heat will denature the hydrogen bond. Its hydrogen bond. It can be denature. Each enzyme function at optimum temperature. For a chemical reaction rate, the enzyme catalyzed reaction generally increase with the temperature within the range in which the enzyme is stable and retain full activity. Right? Most of the enzymes are inactivated at the temperature above 45 to 60 degree. The usual increase in the reaction rate with temperature, the increasing rate of thermal denaturation on the enzyme above the critical temperature. Okay. Some enzymes are thermophilic bacteria. For example, the enzyme of a various species of thermophilic bacteria inhibiting the host springs which are still active at the temperature above 85. Some enzymes like ribonuclease lose activity on heating but quickly regain it on cooling. Okay. If cold temperature, also the effect on the enzyme activity. If the cold temperature is maintained, then the slow down the enzyme activity and decreases the molecular motion of the enzyme. Next is the effect of pH. Effect of pH is each enzyme has an optimal pH that helps maintain its three-dimensional shape. Right. For example, there is show in the graph the pepsin is constant at pH 2 uh, 1.8 uh, something, and the trypsin is near about the neutral 7 to 8, in between the 7 and 7 to 8. Enzymes are markedly influenced by the HIN concentration of the solution. Some enzymes act best in acidic solution, other require alkaline solution. Still, other do not function well unless their environment are neutral in the reaction. These exist for every enzyme: maximum, minimum, optimum pH. The pH value varies with temperature, types of concentration, types of buffer, or presence or absence of inhibition or activators, etc. Some enzymes have a characteristic pH